Altered human consciousness, like ego dissolution, doesn't begin with brain-wide failures. It's triggered by a hyperspecific, microscopic chemical binding event deep within the synaptic cleft. Recently, these same hallucinogenic compounds have demonstrated significant therapeutic efficacy in treating severe, treatment-resistant psychiatric disorders, including severe depression and anxiety. Traditional pharmacology struggles to explain exactly how this works. Most modern psychiatric medications target a single isolated receptor to modulate behavior. Psychedelics do not. To understand their clinical viability, we have to map their cascading biological effects across multiple scales of human anatomy simultaneously, starting from the behavioral level and drilling down to the nucleus of a single cell. This diagram maps a psychedelic event. Subjective experience relies on set and setting. Macro-scale brain network shifts, like reduced CSTC gating and relaxed predictive beliefs, underpin this. Locally, psychoplastogens drive synaptic growth and dendrite complexity. This synaptogenesis originates at the molecular level via receptor signaling. The therapeutic power of a psychedelic cascades from the bottom up. It begins by disrupting massive, established neurotransmitter highways. This diagram shows the physical distribution of the foundational neurotransmitter pathways in the human brain. Classic psychedelics primarily hijack the serotonergic system. These neural pathways originate in a cluster of brainstem nuclei called the raphe nuclei, before projecting densely outward across the entire cerebral cortex. This intense chemical activation specifically bombards the ventromedial prefrontal cortex, or VMPFC. The VMPFC is an anatomical region critical for processing risk, regulating fear, and modulating severe depression. The downstream effects of this serotonergic surge do not stay isolated. The activation forces heavy secondary modulation of both the dopaminergic and glutaminergic transmission systems, effectively rewiring global communication between brain regions. This cascading disruption of major pathways sets the environmental stage in the brain, but the actual physical structural rewiring occurs deep inside the individual neurons themselves. We can observe the exact moment of activation when a psychedelic molecule physically binds to a postsynaptic receptor. This schematic maps the dense, interconnected transduction cascades triggered at the postsynaptic membrane by these compounds. We start with the classic hallucinogenic trigger, the activation of the 5-HT2A receptor. When a compound like LSD achieves high-affinity binding here, it activates both G-protein and beta-arrestin signaling pathways. Beta-arrestin is a protein that regulates signal transduction, plunging the chemical message much deeper into the cell. This pathway initiates a kinase cascade, a sequence of modifying enzymes that ultimately drives straight toward the nucleus, where it dictates new gene expression and commands dendritic growth. While 5-HT2A initiates the classic subjective psychedelic response, mapping the full cellular biology reveals it does not act alone in rewriting neural circuitry. There is a parallel signaling engine operating alongside the serotonin receptor. This is driven by BDNF, or brain-derived neurotropic factor, acting on the TrkB receptor, tropomyosin-related kinase E. Psychedelics bind directly to these TrkB dimers, stabilizing their conformation within lipid rafts on the cell membrane to significantly extend their signaling duration. This extended stabilization triggers internal calcium mobilization, driving through the crucial ERK and mTOR pathways down to the nucleus. This is the vital convergence point in the cell. Both the 5-HT2A pathway and the TrkB pathway culminate right here, in the phosphorylation of the CrEB transcription factor. CrEB stands for CAMP, calcium response element binding protein. Activating CrEB rapidly transcribes new genes. These drive new dendritic growth, express AMPA receptors for neurotransmitters, and ensure overall neuron survival. This TrkB-driven transcriptional output is the measurable biological engine powering the long-term antidepressant benefits of psychedelics. Specific psychedelics like N,N-dimethyltryptamine, or DMT, also activate an entirely separate, specialized intracellular mechanism to support this rapid cellular growth. DMT acts directly on the Sig1R, or sigma-1 receptor. This receptor is located at the critical intracellular junction where the mitochondria meet the endoplasmic reticulum. When DMT binds, the receptor functions as a molecular chaperone. It physically increases the flow of calcium from the endoplasmic reticulum into the mitochondria, which drastically boosts the production of ATP, adenosine triphosphate. This mitochondrial action provides the massive spike in raw cellular energy required to fuel the extensive, calorie-dense synaptic rebuilding ordered by the nucleus. Understanding these exact microscopic mechanisms forces us to confront the primary logistical and medical barrier preventing the widespread clinical adoption of psychedelic therapy. For decades, medical application has been stalled by historical concerns regarding hallucinogenic neurotoxicity and the intense, unpredictable psychological burden of the acute psychedelic trip. In individuals who carry a genetic predisposition to schizophrenia or bipolar disorder, the intense 5-HT2A activation carries a severe risk of triggering permanent psychotic episodes. There are also rare but documented risks of hallucinogen persistent perception disorder, or HPPD, where visual disturbances and psychological distress linger long after the drug has cleared the patient's system. Because of these profound psychological risks, current clinical treatments require extreme, highly controlled medical supervision. This intense resource requirement bottlenecks patient access to the neuroplastic benefits occurring at the cellular level. But researchers are now testing a hypothesis that could bypass this bottleneck entirely. The therapeutic benefits of neuroplasticity may not actually require the hallucinogenic experience. This is the mechanistic decoupling theory. It suggests that the essential brain rewiring, driven by the TrkB and BNDF pathways, is chemically and structurally separable from the subjective hallucinations mediated by the 5-HT2A receptor. Based on this specific cellular mapping, pharmacological researchers are currently modifying the molecular structures of known hallucinogens to create novel engineered compounds like iso-DMT and tabernanthalog. These next-generation analogs are designed to retain the ability to bind securely to TrkB and stimulate rapid dendritic growth, entirely without triggering the 5-HT2A cascade that causes hallucinations. By mapping and mastering the exact cellular neurobiology of these compounds, modern pharmacology is moving closer to isolating the profound physical healing potential of psychedelics from their greatest behavioral risks.